Welcome to Immunovia's live webinar titled Differentiating Pancreatic Ductal Adenocarcinoma from Individuals with Symptoms Suggestive of PDAC, including Type 2 Diabetes with ROC AUC values above 0.95. Thank you for joining us. Before we get started, I would like to take a moment to acquaint you with a few features of this web event technology. At any time, you may adjust your audio using any computer volume settings that you may have. On the right-hand side of your screen, you will see the text chat window. There is a large window which holds all of your sent messages and a smaller text box at the bottom where you will type in your questions. To send a question, click in the text box and type your text. When finished, click the Send button. All questions that you submit are only seen by today's presenters. Your questions will be responded to in the order in which they were received and will be addressed at the end of the presentation. We are joined today by our speakers Linda Mellby, Immunovia's Vice President of Research and Development, and Dr. Thomas King, Immunovia's Medical Director. At this time, I would like to turn the microphone over to our first presenter, Linda Mellby.
Thank you, Megan. I will start with the agenda of today's webinar that covers 4 major topics. The first one is the IMMray technology, and I will take you through the key features of the IMMray technology that really allows us to get valuable information from blood samples. And as a next point, we have shown the ability by using this platform to detect early-stage pancreatic cancer. And the data supporting this was published in Journal of Clinical Oncology in August 2018. And following that important article, Dr. Thomas King, our medical director, will continue to go through the recent results from the optimization study. And here we took a really important step forward, but covering a broader range of control samples, and that are control samples being symptomatic individuals that actually mirrors the clinical setting and reality that we are facing. So we will show some results from this study and then continue with point 4 and 5, our next steps. And that is our next steps in terms of important milestones toward the launch of IMMray PanCan-d, and also the status of our ongoing prospective clinical validation studies. So for the IMMray technology, it's a multiplex proteomic platform. And it allows us to measure the immune response in a blood sample. And the immune response is really the first to react upon a change in the body, such as a disease. And by measuring the analytes within the immune response, we have the ability to detect the changes that occur due to the disease very early on. And also, due to the fact that we are using serum as a sample format, we do not need an advanced tumor mass to detect the disease. We have the possibility to detect it in an early stage of its progression, and that is really an advantage when aiming for early diagnosis of diseases. So in the 2 coming slides, I will briefly explain the IMMray technology, and I have divided it into 2 parts. First, the production of the IMMray microarrays, and then patient testing in clinical laboratories. So our production facility is located at Immunovia in Lund in Sweden, and we will produce IMMray microarrays and ship them to clinical labs for patient testing. And one of our key assets are our antibodies. We produce them in-house, and it's only the binding part of an intact antibody that is produced. And these antibodies are derived from a really large antibody library with more than 10 different variants. And this antibody library really provides us with a fantastic possibility actually to continuously selecting antibodies towards interesting target analytes. So during the production process, these antibodies are printed onto a solid support, and that is the microarray, using an automated robot printer. And the printer puts 1 antibody per spot in a defined pattern, and that is really an automated process. And these antibodies then act as catcher molecules for the specific target analyte. And these produced microarrays will typically contain 10 to 35 antibodies, and that is our biomarker signature. And that also means that we are measuring all these analytes at the same time for each patient sample. And that is what makes this a multiplex analysis, and it's really the information that we got by combining these markers that makes it a strong separation between, for example, disease and healthy. And we can analyze 14 samples per microarray. So once these microarrays are produced, they will be packed and shipped to clinical labs for patient testing. In the clinical lab, you will have your patient serum in a small volume, less than 100 microliter is needed. And the serum has been pre-treated before you apply it to the IMMray microarray for analysis. And as a readout, we use a standard fluorescent scanner. And the signal intensities that we got from the antibody spots will then reflect the analyte binding from the patient sample. Our IMMray evaluation software DX is a cloud-based software, and it will translate these IMMray images and the signals into bioinformatic algorithms. So in the end, the final readout from the software will be a qualitative result, and a positive result will suggest a further clinical investigation. And this whole process of patient testing will take 3 days in total. So altogether, the IMMray platform is a generic platform in that sense that it could really be used for several different diseases. And the only difference is which antibodies you have on the microarray and the bioinformatic algorithms. So after we have launched the IMMray PanCan-d product, we really have a fantastic possibility to develop pipeline products using this platform. But as you know, our main focus right now is really the IMMray PanCan-d test for pancreatic cancer. And in August last year, we published data from 2 large clinical studies on pancreatic cancer in a top-ranking scientific journal, the JCO. And these studies covered more than 1,700 samples from both Scandinavian and North American patient cohorts. And the data showed that we could differentiate early stage pancreatic cancer, and with that we mean stage 1 and 2, from healthy controls with a ROC AUC value of 0.96. And our C value of 0.96 is extremely good. And to put it into perspective, a ROC AUC value of 1 means really a perfect separation. It's 100 specificity, 100 sensitivity, no false positives, no false negatives. Whereas then a ROC AUC value of 0.5 is really the same as guessing. So 0.96 is really a good value for early stage 1 and 2 pancreatic cancer. And the PCA plot to the right in the image shows a clear separation between healthy individuals, which is blue dots, and PDAC stage 1 and 2, the red spots. So these results that we have presented in the GCR article are very important to demonstrate both the potential of the IMMray platform as such, but also the IMMray PanCan-d test to separate pancreatic cancer from healthy controls. So in the next large clinical study that we will present today, the optimization study, we took a step further towards the clinical reality, but by also including symptomatic individuals as controls. So with that being said, I would like to hand over the word to Dr. Thomas King to continue. Thank you.
Thank you, Linda. This is Dr. Tom King. I'm a board-certified pathologist who's been practicing in the United States for the last 30 years. In that role, I've directed clinical laboratories in academic medical centers, as well as in private hospitals, as well as research laboratories in biotech. Today, I'd first like to describe the results of our PanCan-d optimization study, which was recently completed. This study included samples from 923 individuals, including patients with PDAC, stages 1 through 4, as well as several sets of controls. These controls included healthy individuals, individuals with symptoms suggestive of PDAC, such as back pain, and who may have pancreatitis, and individuals with diabetes mellitus. The study is designed to pave the way for our definitive test model building study, which will create the final signature for our PanCan-d assay. This optimization study has several important goals. First, we wanted to extend the work that Linda has already described for you by assessing the ability of the IMMray platform to distinguish between patients with PDAC and those with symptoms suggestive of PDAC who actually have another type of GI disease, such as pancreatitis. We felt this distinction is very important in terms of how this assay will ultimately be used in the clinics in terms of evaluating patients with suggestive symptoms who may or may not have PDAC. This is really an ideal indication for a non-invasive blood test such as IMMray. Second, it's very important for us to evaluate IMMray's performance in diabetic patients, as we feel a major indication for this test will be in individuals with new onset diabetes after 50 years of age. Such individuals have a fairly substantially increased risk of PDAC in the first few years after their diagnosis of diabetes. So assuring that this assay performs adequately in individuals with diabetes mellitus is very important to us. The third goal of this study is to assure that a single IMMray signature can distinguish between diabetics and symptomatic patients as well as healthy controls. This is important for us because a major indication for our test will be individuals who are healthy but are at genetic or familial risk for developing PDAC, with very substantial PDAC risk over their lifetime. Finally, this study evaluated the value of adding CA19-9 results to our IMMray PanCan-d signature. And as you probably know, CA19-9 has been used as a serum biomarker in patients with pancreatic cancer for many years. It's useful in following the progression of disease, but it lacks the sensitivity and specificity that's to really make it a useful screening test for detecting early-stage pancreatic cancer. We felt that there was a reasonable possibility that adding this to our signature could enhance the sensitivity and specificity of our assay. This shows the results of the optimization study with separate area under the curve analysis for PDAC versus healthy controls, individuals with diabetes mellitus, and symptomatic individuals who may have pancreatitis or other types of GI disease. This area under the curves are shown in conjunction with CA19-9, which has been incorporated into the IMMray signature. In all three of these cases, the area under the curve is very high, greater than 0.95, ranging from 0.96 to 0.98. And as Linda indicated to you, this shows a very high level of discrimination between PDAC cases and each of these controls with a single signature. This then really verifies that the optimization has developed a signature that works very, very well in a more realistic clinical situation than prior studies. In addition, we found that CA19-9 added approximately 5% area under the curve increase, so that it seems to be very valuable for our assay. And we intend to include CA19-9 results in our test model building study, which is beginning now. Another important finding from this study, which again supports Linda's previous results, is that low-stage PDAC, stages 1 and 2, or easily distinguish from all of these controls using a single signature. This is very important, as we hope our test will be able to identify individuals with potentially resectable pancreatic cancer. So being able to detect these early stage cases with a high level of sensitivity and specificity is very important for us, and we feel very optimistic that this will be a very important clinical test. This optimization study has also allowed us to refine our selection of relevant antibodies and optimize their concentrations in preparation for our next step, which is the definitive commercial test model study. As we said, this is beginning now and should be completed by the end of the calendar year. As Linda has told you, there's substantial literature based on important prior studies in multiple populations that provides our rationale and plan for PanCan-d development and commercialization, and that is what we are implementing now. The next step is this large commercial test study, which will involve up to 1,200 samples that are freshly collected, that is, collected within the last 2 years, and these have been obtained from our collaborators in centers around the world. This study will complete by the end of 2019 and will yield a locked definitive antibody signature for the final clinical assay. Once that has been completed, the next step is a verification of that signature, and that will include 600 independent samples that will be assayed with the locked signature. After the verification, we will proceed to a final blind clinical validation that will involve an additional 600 samples that will be received with no knowledge of the individual's PDAC status. This final validation will then clear the way for us to launch this test in our U.S. laboratory as a laboratory-developed test at the IMMray DX laboratory in Marlborough, Massachusetts. This slide summarizes the milestones that I've just described for you. The commercial test model study should complete by the end of this calendar year. In Q1, we'll begin the verification study, which should complete in Q2. And in Q2, we will do the final blind validation study that clears the way for us to offer the test in the United States. And then we plan to launch in Q3 at our IMMray DX laboratory in Marlborough, Massachusetts. Concurrent with this test development, we are continuing 3 large prospective trials that will eventually involve more than 10,000 subjects. These clinical trials are designed to support the main indications for our test. The first is PanSim-1, which is being conducted in collaboration with University College London and looks to collect more than 2,000 samples who have screened for symptoms suggestive of pancreatic ductal adenocarcinoma. We intend to prospectively evaluate these samples with our assay and assess its clinical performance in this important setting. The PanFam-1 study has been ongoing for several years with collaborators around the world. We expect interim analysis of this in 2020. This study is designed to assess the effectiveness of our assay in individuals at familial or genetic risk for pancreatic adenocarcinoma. Finally, the PanDIA-1 study is more recently initiated. This aims to look at the ability of our test to identify PDAC in individuals with new onset type 2 diabetes mellitus after the age of 50. And this is being done in collaboration with Swedish centers. And we expect more than 6,000 samples from this. All of these will have an interim analysis in 2020 once we have the final signature for the PanCan assay, and we expect final readout from these in 2022 or 2023. These collaborations have also allowed us to develop strong relationships with many key opinion leaders who are based at these institutions, and we feel that they will be helpful for us in terms of launching our assay clinically. Finally, to summarize, The successful optimization of the IMMray PanCan-d assay in combination with CA19-9 produced area under the curve greater than 0.95 in differentiating PDAC from symptomatic patients, diabetics, and healthy controls. Our remaining steps to market are the commercial test model building, a verification, and a blind validation. And all of these are on track for us to launch in Q3 2020 at our IMMray DX laboratories in Marlborough, Massachusetts. Our prospective clinical studies to evaluate and support all of the major indications for this test are ongoing and on track, and we expect an interim analysis in 2020. We are aiming to collect more than 10,000 blood samples in these trials that will give us the necessary evidence for FDA clearance and to enhance national reimbursement. Thank you for your attention today, and we'll now take questions.
We will now begin the question and answer session. As a reminder, text chat is located on the right-hand side of your screen. To submit a question, type your question in the small text box at the bottom, and when finished, click the send button. Please note that due to time constraints, our panel may not be able to respond to all questions submitted. If our panel is unable to address all the questions submitted during the webinar, then those questions will be addressed individually after the program.
Hi, this is Julie Silver. I'm Director of Investor Relations at Immunovia, and I am going to be the question asker. So those of you who've dialed in your questions, I will do my best to ask our panel the questions that you want to hear from. The first question we got is, when we receive a positive result from IMMray PanCan-d, will it mean more clinical follow-up? What does it mean for the words further investigation?
And I'll take that question. I'm Dr. Tom King. The diagnosis of pancreatic cancer is difficult, and it requires multiple modalities in order to do this appropriately. NCCN guidelines recommend that these decisions be made in a cancer center that performs many Whipple operations and has a multidisciplinary team of surgeons, pathologists, radiologists, and others who are expert in diagnosing pancreatic cancer. The IMMray test will be in addition to these modalities, and we feel that it will provide very useful and in many ways more sensitive and specific information than those that are currently available, such as imaging.
Thanks, Tom. So our second question is about the labs. Does the lab in the U.S. look and function like the lab we have in Sweden?
And I'll take that one as well. I'm the medical director of the laboratory in Marlborough, Massachusetts. We're a bit farther along in that laboratory than we are in the one in Lund, Sweden. This is because we anticipate launching the IMMray test in Q3 2020 in Massachusetts. That will be somewhat later launch for the European version in Lund because CE mark is required before that test can be launched there. We're launching as a laboratory-developed test in Marlborough, so we can be a little bit ahead of the curve. And in order to accomplish that, we have a very nice facility. Most all of the equipment is in place and has been validated, and we're ready to move forward as soon as the commercial test model building study is complete.
Thanks, Tom. So we have a question here about what kind of regulatory environment will the PanCan-d be subject to?
I'll take that again. In the United States, we plan to launch IMMray as a laboratory-developed test. This is a pathway that can be taken by high-complexity laboratories that are usually inspected by the College of American Pathologists. In that mode, we do not require FDA clearance to proceed with the assay, and we intend to seek both CLIA and the College of American Pathologists accreditation as soon as we launch the test. This is a modality for testing that many new types of tests employ. I personally have initiated many laboratory-developed tests using PCR-based technology to diagnose both infectious disease and lymphomas and leukemias. So I think we have very good expertise in terms of managing the launch of this type of test. In Europe, the pathway is slightly different. A CE mark is required before the test can be launched clinically. So that will delay us until that is complete. We do anticipate seeking and gaining FDA clearance as soon as we have completion of our prospective studies.
Great, thank you. So we have a question here that basically is asking, will we start our larger studies over again now that we know the extra value of using CA19 biomarker?
The answer to that is no. What we've done is to prospectively collect samples. We have not completed the analysis of those yet, and we will not do the interim analysis until we have a locked signature, which we anticipate will include CA19-9.
Great. When and where will the first out-of-pocket test be sold?
The first out-of-pocket test will definitely be sold in Marlborough, Massachusetts. Our plan is to offer a self-pay model for testing to individuals who are at familial and genetic risk for pancreatic cancer. We've identified many of those through the cancer walks, through the important organizations of patients supporting individuals with pancreatic cancer and lobbying for earlier diagnosis of this disease. Making contact with these individuals at many of these cancer walks across the country, we've been able to talk with them, and many of them are very interested in being tested. And we think that these will probably be our initial patients.
Great, thank you. Can you elaborate a little bit on the launch plans for both the U.S. and the European markets?
I can take it on. This is Laura Kika, and I'm in charge of the commercialization. So we have been working intensively with the market access and also very much in detail with the launch plans, and both for the LDT launch, as Tom was just explaining earlier, will take place towards Q3 next year. We have identified all the different events that we will participate in. We have set up all the different channels, the collaborations with the key opinion leaders. We have a fantastic network through our collaborators in the prospective clinical trials that we have been working for several years now, and they are also guideline authors, national guideline authors in our prioritized geography. So they will be helping both for the U.S. launch and for the European ones. And we are pending in Europe the ISO 13485 certification, and once that is going to be in place, we will, of course, obtain our CE mark IVD for IMMray PanCan-d for sales in Europe. So we have extremely detailed plans for our prioritized geographies, both US and Europe.
Thanks, Laura. Okay, so our final question is about the webinar series and how many more of these webinars can our audience expect, and when we might be able to share more information.
Yes, so this idea actually with the webinars we like very much because I think it's a very good way to communicate our results, and we have decided to create a series of webinars that will in most of the cases will follow our milestones. So now we have achieved a major milestone with our optimization study results, and thereby we have our first webinar. And you can also find the supporting data in the poster that you can find on our website, and that has been presented and will be presented at several conferences throughout this autumn. And then
as soon as we are going to have the commercial test model results, we are going to announce those results and present them in a webinar as soon as they are available towards end of this year. Then we are going to move on with the verification study, and once we have those results, we will be also communicating that. And of course, very important, the blind validation and our LDT launch will also be included in webinar. And moving on, we will also be having the interim results from our, as Tom was mentioning, more than 10,000 individuals that are included in the 3 risk groups on the hereditary, symptomatic, and also new onset diabetes. We will have those as soon as we have a validated test that will measure all these 10,000. And then moving on also, as soon as we are getting more results from the interventional studies that are the next stage that we are moving on with the prospective clinical trials, we will be sharing those as well. So we are actually looking forward and planning a large number of these events.
Great. Thank you, Laura. I just want to remind everyone, if they want to ask a question, to type it into the browser and we can ask it. We have one more question that just came in. Can you elaborate on the pros and cons with proteomics, since most competitors are using a ctDNA technique?
Well, ctDNA-based assays depend on DNA released into the bloodstream from tumor cells that have died and release their DNA. Usually, although these assays can be sensitive in terms of the number of molecules they can detect, it usually requires a fairly large volume of tumor in order to release sufficient DNA to be present in a small blood sample which is analyzed. For this reason, we feel that many of the DNA technologies are really insensitive to very small tumors. They may be good at identifying tumors at a later stage of development, but are not very good at identifying very early stages. Proteomic assays depend not only on products of the tumor, such as CA19-9, but also on the host's immune response to the tumor, and I think very relevant for PDAC, the stromal changes that develop within a tumor and the products of those stromal cells. So we feel fairly confident, certainly based on our optimization study showing an area under the curve of 0.96 for early stage 1 and 2 PDAC, that we have an assay that is very, very good at detecting early stage PDAC. I think it would be hard for a DNA-based assay to mimic this. So we feel confident that our approach is a rational and I think will be a very successful method for identifying early stage disease.
Thanks, Tom. If we have no further questions, I would like to thank everybody for tuning in to this webinar series and to stay tuned for future events and future news that we will be sharing on a regular basis. It's really great to actually have the whole team under one roof. We're very excited to all be together here in Sweden, and we're very, very pleased that you were able to join us. Thank you very, very much.